welcome to What's Up Life Science, a series where we invite voices from across life science ecosystem to share with us their journey and story. And today we are going to focus on AlzeCure Pharma. I have with me Martin Jönsson, the CEO. Martin, very welcome.
Thanks so much.
Thank you for being here.
Thank you.
You are working in two areas, both the Alzheimer's disease and pain.
Correct.
Could you go back and give us a little bit of the history, how the company became into life?
Absolutely. So AlzeCure Pharma was founded here in Stockholm and here in Flemingsberg. And we are a spin-out out of AstraZeneca. So when AstraZeneca left the CNS space, AlzeCure was created. And that with the help of the Swedish Alzheimer's Fund and with Professor Bengt Winblad at Karolinska Institute. Initially, we were a research foundation, but very quickly the team had several projects where we really saw meeting a very big unmet medical need. And thereafter, the company became a traditional company which did an IPO. So in 2018, we went on the stock market and are listed at Nasdaq, First North Growth Market.
And how big is the market for Alzheimer's disease if you only focus on that?
It's very big. I mean, today there are about 50 million individuals who are suffering from dementia. And the patient population is expecting to increase with 300% in the coming 30 years. And this is due to the aging population, but also that we are living longer and longer. And today there are no curing treatments for Alzheimer's. And there's a huge unmet medical need. There has been breakthroughs, but these breakthroughs do not meet all the needs in the market. So there is a huge unmet medical need. And also that people who get dementia and Alzheimer's, they are in different states, so you need to maybe then go in and see to it that the development of Alzheimer's is getting slowed down. But then the individuals also have cognitive deficits, and then you need to go in and improve the cognition. And
but you're not only working on slowing down the disease progress, also you're trying to improve the cognition.
Exactly. So we are working on both improving cognition, but we're also working on preventing that you are developing Alzheimer's, because individuals who are developing Alzheimer's is doing it over a time period of 10 to 20 years. And initially, you don't have symptoms. But what's happening is that you are producing more and more toxic proteins, amyloid beta 42, which are building up in the brain and breaking down the brain. And with our therapies, like alstatin, we should be able to go in and identify and see from a blood sample, are you producing more of these toxic proteins? And then also doing a gene screening and seeing...
So we already have like the biomarkers and gene predictions?
Yes, absolutely, absolutely.
So we can already know if I'm going to get Alzheimer in future?
We can know if you have a higher probability of developing Alzheimer's. What's great is actually that some individuals who are developing this pathology, they are not developing Alzheimer's while they live, but if they would live long enough, they would surely develop these problems.
Yeah, I was actually visiting your lab and some of your experts, employees, and I would like to show our viewers what I saw in the lab.
Absolutely.
So let's take a look.
Yes, let's do that.
One of the very interesting companies in our life science cluster in Flemingsberg is AlzeCure, which is working in the field of Alzheimer and pain. And I'm going to meet Johan Sandin, their Chief Scientific Officer, to get an understanding of what it really means to work with Alzheimer's disease. Hello,
Hi.
Johan. Thank you.
Good to see you. Welcome to AlzeCure Pharma.
Thanks a lot. So I have read a lot about AlzeCure,
Yeah.
but I have always wondered, what does it really mean? What are you doing in the lab? So I thought it's a good idea to take a look at it, if you can show me something.
Absolutely. So we're working both with cell biology, molecular biology. Well, we do a lot of chemistry as well, in vivo biology. And this is one of our labs we're working with. Here we have general molecular biology going on in here. So there's a lot of experiments, and I'll show you some other experiments going on later on. Here we also have cell biology labs where we do a lot of the in vivo cell biology ourselves. So in here we are working with a lot on imaging. So here we have an automated microscope where we can look at various types of cells, for example, in real time, and look at how they respond to various drugs.
So are these
yes, so here,
neurons?
Yes, so here we have neurons. So these are nerve cells in live, and we can see there the cell bodies, and you can see the extensions, so-called neurons, which they make contact with other nerve cells. And here we can see how our compounds stimulate how these neurites grow and make contact with other nerve cells. So that's really important for our compounds, that they stimulate nerve cells which are dying off in Alzheimer's disease. So that's one of the things we're working on.
And these are live? And,
yes, exactly.
Where do you grow this?
So we grow these in plates. So these are various types of plates that we use. So these are ordinary 6-well plates that we can grow them in, but we can certainly grow them in 96 or even 384 well plates. So we can actually make 96 or 384 experiments in one plate.
And what happens next?
So next, we obviously want to see what is driving the mechanism behind the neural outgrowth. So then we turn to Cristina, which is working on the other side with more on the mechanism of action, if you will.
Hello, Cristina.
Hello.
Sorry to interrupt you.
It's okay.
What are you doing?
So we are exploring the mechanism of action of ACD856, and we are today evaluating in both kinase array and Western blot. So we have cells in the cell lab that overexpress TrkB receptor, and we incubate with the compound, and then we look at the activation of multiple signaling downstream the receptor using these membranes where we can screen multiple signalings. So we detect the specific sites of these proteins that are phosphorylated. And based on these results, where we detect the proteins, we move to Western blot, and we can confirm the data using same samples, comparing across the treatments with compound. And what we do here is basically that we load samples, separate by size, the proteins, and then we detect with primary and secondary antibodies by using chemiluminescent.
Thank you so much. Thank you. I'm sorry I interrupted your work. Good luck.
Yeah, thank you very much.
Johan, you told me you have a mass spectrometry instrument as well. Is it possible to take a look at that as well?
Absolutely. So let's go and have a look. So in here, we have our mass spectrometer. And this is an instrument where we do analysis and look at how much of the drug we're making we can find in the blood of the animals that have been treated with it. So we can look at exposure here, we can look at the purity of the compound, etc. So this is a really useful instrument, and here you can see various peaks. So this is, for example, a peak of our specific compound, looking at it, seeing that it's pure, and we can also look at the calibration curve of the compound here. So this is an instrument we're using both for pharmacokinetic analysis, but also in really integrated in our daily work.
So how often is it being used? Is it all the time?
It's being used most of the days in the week, actually. So this is a really used instrument.
Thank you. So now I ask Johan to show me more concrete, what is it they are actually treating?
Yes, so we picked this brain apart a little bit, but the area we are trying to affect is the hippocampus, which is affected very early on in Alzheimer's disease. And that's the area that starts here and goes down in the kind of C-shaped here. And you can see the projections actually connecting to other areas of the brain. And it's an area which is dying off very early on in Alzheimer's disease. And it plays a critical role in memory. That's also why memory is affected very early on.
And that was because of the amyloids?
Yeah, because of the amyloid plaques that's forming in the brain.
That was great. So you have actually a big pipeline with 4 different candidates.
Correct.
Let's start with NeuroRestore.
Yeah.
Which phase are we in and what are the coming milestones?
Okay, so with NeuroRestore, where we are focusing on Alzheimer's disease to improve learning and memory capabilities, we are going into phase 2. And NeuroRestore is a project that has EU funding. So we got funding last year from the European Innovation Council, who are going to support our startup of our phase 2. And the plan is to start that project this year.
They were also looking into doing some investment.
That is correct, yes.
Because you got the fund, which was 2.5 million euro, which is really huge because only 2% of applicants get this fund. And you were the only Swedish biotech company getting the EIC fund, right?
Yes, we were. We were the only Swedish biotech pharma company who got funding last year. So that was a great accomplishment. Kudos to the team.
And if we look at Alzstatin, what does that meant to do?
Alzstatin is focusing on that as we get older, we are producing more and more toxic proteins, amyloid beta 42, and these toxic proteins are breaking down the brain and creating plaque. What we are focusing on is going in the best case early to see to it that we lower the production of toxic proteins so we do not harm the brain to the extent that you develop dementia. And then also to go into patients with Alzheimer's and treat them to bring down their load of amyloid beta 42, this toxic protein.
And I've read that you can also use your candidates in other indications like Parkinson and probably even depression.
When it comes to NeuroRestore, many of these patients who have neurodegenerative diseases, they also get cognitive deficiency. So we see that we can go in and treat these groups. Also in depression, we also see that the mechanism that we have, increasing the signaling in the neurons and increasing the levels of BDNF, brain-derived neurotrophic factor, is a potential treatment of depression. So we know that the existing drugs like ketamine, psychedelics, SSRI, that their antidepressant effect is through the TrkB, the BDNF receptor. So that is also what we are going after.
Now that you mentioned BDNF, and I know you yourself do a lot of sports.
Correct, yes.
And we get a lot of this BDNF through doing sports,
Correct.
right?
Yes.
So can we say you are having a pill, like the exercise pill, being created?
Some actually say that that's the case, because when we look at in animal models, we can see that we can increase the levels of BDNF after 28 days with around 30%. But we should never forget that when you exercise, there are so many other benefits that you are getting.
And if we go back to the pipeline, now we have talked about the Alzheimer's disease, but you have also pain.
Correct.
Could you elaborate a little bit on the 2 candidates?
Absolutely. So it's fascinating because, you know, when we went into the Alzheimer's field, we were looking for molecules which could increase the neural signaling. And as we were looking for these molecules, we actually also found molecules which were blocking the neural signaling. And we are using some of these molecules to block pain signaling. And one of our programs is TrkA-NAM, where we focus on pain and osteoarthritis. So we are blocking off the NGF signaling. And this NGF signaling is very central when it comes to certain pain disorders.
What are the alternatives for that today?
They are very bad. And so to a very large extent, opioids are being used. And in many cases, the people who are getting treatments are not experiencing that they are getting an adequate pain relief and often need to go to opioids. So there's a very large interest to find alternatives to the opioids.
And for the ACD440, which actually comes from AstraZeneca, you got some positive reaction from FDA,
Yes.
you got the orphan designation.
Correct.
What does that mean for AlzeCure?
It means a lot because the recognition from the FDA, who gave us orphan designation for ACD440 for the pain disorder erythromelalgia, and also they gave us positive feedback on a potential pivotal trial. That's a great, recognition of the program, but also on the trials and the material that we have developed. And what's also great is also that we have received orphan designation from the European Medical Agency, EMA. So we got that a couple of months ago. So the interest around this project has really increased, and it's a recognition of the program and the abilities of the company.
So why are orphan indications so attractive?
Because often indication, they have several different pros. Often you can do smaller trials, which leads to smaller investments. And you can also drive, in several cases, these programs quicker. And then when you get to market, you get market exclusivity in the US for 7 years, in Europe
Which
for
is
10
normally
years.
5 years, right?
Exactly, depending on the molecule. So that is very attractive. And then you also get tax benefits around when you do the trials. You can also get quicker advice. And you, as a biotech as we are, you can also get free advice. So there are several different benefits. So it's a big advantage for us to have that.
Now you reminded me of a patent, which is as important as the science. So how is the patent portfolio looking?
First of all, I would say that the patent is almost more important because if you don't have strong patents, you will not have money so you can develop your programs. And we are very happy that our situation is very good. So on our Alzheimer's program, Alzstatin, we filed last year and we are now preparing for phase 1. On new restore, ACD856, which is going into phase 2. There we also have a very good patent situation, already granted in all the big markets in US, Japan, Europe, China to 2039. We have just submitted a new one, which would give us additional 7 years. On TrkA-NAM, also on the lead compound there, filed last year. On ACD440, we also have patents to 2042. So we are working very consciously on patents and patent strategies and having several different patents. So we actually can go after several different indications and out-license them to several different companies.
So now about the out-licensing, what is the business model? Are you focusing on selling, on licensing or partnering?
Our strategy is that we should out-license 1 to 2 of our programs, and we should take them the furthest to phase 2, and through the out-licensing, getting financing for our other programs. We see that we have our strongest competence in early discovery, research, and early development. So our aim is to out-license to major pharma companies who can do the last trial and also do the commercial launch.
And we know many companies during the past years have failed to work in Alzheimer.
Yes.
So what makes AlzeCure different?
Yeah, the thing is, now with the additional knowledge we have in the Alzheimer's space, where we, to much better extent, can identify which patient is it that has Alzheimer's, because in many of the previous studies, actually the patient who were included didn't have Alzheimer's. And now we also understand the disease much better. So going after utilizing the science that has now been proven, we are also increasing the probability of success. And for us, with Alzstatin, going after the buildup of these toxic proteins and also going after increasing the neural signaling, we have very good scientific backing, and we're also working with several of the core global thought leaders like Henrik Zetterberg, Bart de Stroper, etc. And we have worked with them for very many years, and they continue to work with us. So that's a validation for what
almost
we're doing.
answered my last question.
Okay.
I wanted to ask you, if someone is looking into AlzeCure, why should they partner with you or invest in you?
Yeah, I would say we have validation from FDA, from EMA, from European Innovation Council, from world-leading key opinion leaders. And based on that, I think we have validation of our project. And we have this validation on several projects, so that is validating the team and are increasing the probability of success. So I think those are good reasons.
Those are very good arguments. Thank you so much, Martin, for being here and giving us your time.
Thank you for having me. Thank you.